So far, we've discussed the passive properties of the neuronal membrane. Let's review a bit about what we've learned so far. The membrane potential for a given patch of membrane is the electrical potential difference between the inside and the outside of the cell, caused by the relative concentrations of charged ions like potassium and sodium inside and outside of the cell, and by channels which allow the flow of these ions down their concentration gradients. The resting potential sits at the balancing point where diffusion and electrical forces are equal and opposite for the charged ions inside and outside of the cell. We know that the fluid inside and outside the cell is basically net electron neutral, and that only a very small number of ions, like potassium, moving across the membrane can cause big changes in the potential across the membrane. Beyond the resting potential, we know a few things about the passive circuit properties of the membrane. For instance, we know that the lipid bilayer acts like a capacitor, and its properties fundamentally limit the speed at which the membrane potential can change. Charging and discharging the bilayer is not immediate. We can also talk about the membrane resistance, another term borrowed from electronics, which describes how much the membrane resists the flow of current through it. The membrane itself is a good insulator and has a high resistance, but it's full of ion channels that allow ions, and therefore current, to pass through. Membrane resistance is governed both by the ease with which ions can flow through the individual ion channels and by the abundance of these channels. So why is all of this important? All of these mechanisms set the stage for neuronal signaling, but everything we've talked about so far is fundamentally passive. So what actually allows us to change the membrane potential and to send a signal? Let's start our investigation of signaling by looking at something a bit more tangible. Globerus discoidalis, a South American cockroach. Let's quickly have a look at some live neurons in a real-life experimental setup. Up until this point, we haven't actually seen what happens in vivo in a living creature. We're using a device called the Spiker Box, a piece of open source hardware from the team at Backyard Brains. If you have one, we encourage you to do this same experiment at home. Now, this is a juvenile cockroach, which we've anesthetized in some cold water. We'll cut off his leg like this. Don't worry, he's not an adult yet, so his leg will grow back. We insert the two electrodes in two parts of the leg, the femur and the coxa. And we're measuring the electrical potential difference between these 2 points. This one is the ground, and this one is positioned right next to a large bundle of nerve fibers, or axons, that run up this little guy's leg. Because our electrode is close to that bundle of fibers, and because there are so many fibers, we'll be able to indirectly measure what's happening in the membrane potential of these fibers. When we put the sound cable from the Spiker Box into a phone or laptop, we can hear lots of popping noises. When we actually look at the sound signal, we can see a lot of noise and occasional blips of spikes like this. If we touch the hairs on the leg, more spikes, and in sync with our stimulation of the leg hairs. Signals. So what are we seeing here? We'll call these blips action potentials, or spikes. Now, technically, these are compound action potentials because we're seeing the activity of many cells firing at once. But the basic idea is the same. We see a huge, rapid, transient change in the membrane potential of these axons. What we've learned so far about the passive properties of the neuronal membrane is not enough to explain what we're seeing here. So we need to introduce some additional machinery to explain what's going on. Let's think about what could allow us to rapidly change the membrane potential, to initiate this change in membrane potential, and to produce an action potential.